washing machine.
Yes.
This is its sole function, is to wash the cells and make sure that any other materials that are in there are rinsed out that are unneeded.
Correct.
Okay, all right. So can you tell me a little bit about what we've got set up here to run our washing machine?
So when we left the other machine, we got our cells of interest. We then spoke about how we would like to count the cells because we need to know what the concentration is in here. And the reason we want to know the concentration of the cells that we're starting out with is for the LOVO, you have to tell the LOVO what the viable cell, you would like to know what the viable cells are, but more so what the total cells are that's going to run through the machine. And the reason for that is that inside, one of the key things about the LOVO is this membrane, and the space between the membrane and the outer plastic is about 10 mils. And you need to know what's about to go into there in terms of the concentrated cells. You need to know whether you need to dilute that, because if it's too concentrated, you'll either foul the membrane and completely clump up the entire system, and you won't get a good wash and concentration of your cells.
So Chesney, now that we have the bag of cells, which are going to be our source, welded in, what are the next steps in actually cleaning the cells up here?
So the next steps is you've set everything into the machine. The machine's very intuitive, so it will tell you when to do things. In other words, when to mix the bag, when you need to mix the bag, when to add something if you need to add something. But most of the time it will just prompt you to mix. And if there's an alarm, if something goes wrong, a pressure alarm of any kind, because the volume's incorrect or something happened, there's air in the bag. So this machine has a bubble sensor.
Okay.
So if you told the machine that there's a specific volume in the machine and the machine pushes it through and less of that volume is in there, meaning air will suddenly start going through the system, the sensor will detect that and will alarm.
Okay.
And it will prompt you that you said there was 500 mils in here, but I'm only detecting 400 mils or something like that.
Okay.
Or it will just tell you that it's less than what you had told it.
So I can see we've got some liquid spinning around in this part of the machine. Can you explain what's going on in here and how it connects to everything else?
Yeah, so this is one of the big things about the LOVO machine is you have the spinner membrane, and most of what happens in terms of separating cells from either washing them happens within this spinner membrane. So what will happen is your cells will come along a path in the system, will travel down into the spinner membrane, which then between the membrane there's a space that equates to about 10 mils across, and then there's an outer wall. In that 10 mils is where your cells will be traveling. So they'll be going in, travel all along, and then they'll come back out here where they'll go through these peristaltic pumps depending on which way the cells are flowing. So usually when the cells tend to flow into the system, they'll come along this path, and then they'll go in the membrane and they'll come through the red path over here. And while the spinning and concentrating is happening, the machine will control which way the cells will be flowing into that. On the other hand, anything such as the media or anything that might be in that in your source bag, like in terms of you wanting to wash it out, that's smaller than 4 micrometers, will go through the membrane to the inner casing of the spinning membrane, and that will travel down at the bottom over here into what's called the filtrate or the waste bag.
I see. Okay, so if I understand you correctly, then that's the cells and all their liquid come through here. The cells, because they're 4 micron diameter, get through the membrane and back.
They're larger
out.
than 4 micrometers.
Larger than 4 microns. So the things that are larger than 4 microns, which are the cells, come out the bottom and go this way.
Correct.
Smaller than 4 micron, because it's just waste, comes out the bottom.
Correct.
Okay.
And if you remember earlier, we said that we were doing 3 cycles, so that process will happen 3 times.
Okay. So that process repeats, and they wash, go through, wash, go through, wash, go through, and I end up with a concentrated cell suspension with all the media over here.
Yeah, through every wash, the cells get concentrated. And a lot of that settings, as I mentioned as well, if you want 3 washes, more washes, if you might want to add another buffer or media in that, can be programmed into the machine. And that can be done by the operator, at least on the PD side, but once that program has been set in a manufacturing suite, it's usually locked in. And early on in the machine, it will ask you choose which program you're going to run. It will ask you for the operator to put in their initials, There might be a lot number or batch code that might have to come into the system as well. And then all these things get logged in the machine. So if any point something goes wrong, if there was a power outage, which shouldn't happen because you're standing here, but could happen, especially in a GMP facility, or some random thing just happened, that will all be logged in.
Okay.
So that will make it easier further down the line if you need to troubleshoot.
So the machine is actually really automated, but there's a lot of interaction between the human and the machine.
Correct.
So what happens if, for example, you're going from one wash cycle to another? Does it tell you? Does it ask you to do anything?
Yes, it does. So you do, as an operator, interact a little bit with the machine. One of the ways that you interact with the machine is the machine will prompt you to mix the bag. So if you have really concentrated cells, they can settle down very quickly, or if the cells are really large. So you will, during the wash and concentration steps, you'll get a prompt to mix the bag. And once you've done that, you have to interact with the machine by saying, yes, I've completed that task, and it will allow you to move on to the next step.
Okay, so why don't we get started washing these cells?
Why don't we? So after you set up the machine, it gives you a prompt. You've done everything. The system is ready to begin the cell processing. So when you click resume, the first thing that it asks you, it says pre-wash cycle, can you please coat the bag? So this is one of the ways that as an operator you would interact. So what that means is it's asking me, similar to priming the line, it's asking me to prime the bag.
Okay.
So it's just mixing the bag a little bit. So usually what we tend to do is just have some of the buffer that has gone through the machine to just wet the bag.
So that way the cells don't stick to the side of the bag.
Correct. Especially since this is a big bag, because you might have a very large volume. You don't want any of your cells to be stuck on top. Again, your cells are very precious, so you want every last drop of them. So I've done that, so I'll put that back on. So then I say, yes, I've done that, so it's resume. And what will happen next is your source cells will start going through the kit.
And that click that we heard was a valve opening?
Correct.
Okay.
So it was opening the source path, and now your cells will go through the source path. And as you can see, it's going through the spinning membrane, and this is where you will have a separation of waste, and your cells will be traveling this way. Sorry, it's going up. Yeah, it's coming this way. And it will eventually go into your in-process bag.
Are we filling it yet? No.
It's slowly working its way up.
So that's really helpful to see. So this is coming in from the source bag, getting washed. The cells that are larger than 4 micrometer diameter go this way. The waste goes this way.
Correct.
So once you're in a wash cycle, how long does that wash cycle last? Can I go watch a television program?
No, it depends on the source, the volume of your source. That will depend on how long it will take. But again, in the beginning of the program, it doesn't break down specifically the cycle time.
Okay.
But it gives you an overall time of how long this process will be taking. And usually, you don't want to go and watch television when you're working with cells. And in a GMP suite, there will always be someone at the machine. You never know when something might go wrong.
And as you said, the cells are very precious.
Yeah. That's it.
This is a perfect example.
Air in the bag.
All right. So here they detected air and the machine tells you.
Yes. So in this particular example, what happened was that all the cells have kind of shifted down to the left, and air went through the system. The bubble detected air. And as you had mentioned, air has been detected. And the nice thing about the machine, it gives you options. First of all, it's telling you what potentially went wrong. You can also click to give you an explanation of why is there an error.
Okay.
So in this case, it's telling you air was detected, and in some cases, it will tell you either to go to the manual or it might give you some solution. So in this case, I'm going to go, okay. And now it's giving me options. Would I like to continue? Is my bag not empty? So I'd like to continue. Do I want to clear the line? Or I said there was 500 mils in there, but maybe there was less than that. Do I want to go to the next step?
Okay.
So in this case, I know there's more, so I can click that. And again, it's always asking you, are you sure? Is everything fine? Click yes. And so this step is asking me who's the operator that's allowing this to go ahead. So it's me.
You're taking responsibility.
Correct. And I'm ready to go, and so we'll continue.
So now that we've got our cells washed and we've got them in a bag, where do we take that bag? What's the next step in terms of turning those clean T cells into a therapeutic product?
So the next step would be a check of the cells. So if you remember, I said we counted the cells in the beginning because we needed to know how many cells were alive, so viable cell count, and how many cells were we actually washing. So the same thing will happen again. At the end, I'll take off my cells. I'll take a sample of that cells in a sterile way, and then I'll count it. That will give me some indication of how many cells I might have lost. Hopefully, I haven't lost much. The machine usually collects you get a very good recovery.
Okay.
Especially when you have when you have a lot of cells, if the concentration was really high.